My name is Ashley Miner, and I'm here today to talk about cleaning and sanitization of salmonella-contaminated peanut butter processing equipment. A little bit of history about myself. I spent some time studying poultry and salmonella contamination, as well as have always found foodborne illnesses to be of interest. So experiment is kind of a combination of the two. A little bit about salmonella. Salmonella is a gram-negative motile bacterium, which is versatile and abundant. It can be found in a variety of animals, including warm-blooded and cold-blooded, as well as people and the environment. It is a facultative anaerobe, which means it can survive with or without oxygen, which only increases how far this bacteria can go. Originally, before the 1990s, nut butter and other low-moisture foods were considered immune from contamination by foodborne illness bacterium, such as salmonella. However, since the mid-1990s, there have been 5 outbreaks of food poisoning caused by salmonella contaminated in peanut butter. The contamination can occur at any point throughout the growing process of peanuts or nuts, and as well as throughout the manufacturing process and packaging point. While foodborne illnesses are not life-threatening, salmonellosis, or the illness that you would get from salmonella-contaminated foods, can cause flu-like symptoms for 4 to 7 days and can lead to dehydration if not taken care of, which can lead to hospitalization. So how do we ensure that our manufacturing processes are not causing the contamination of salmonella? Traditionally, there are 2 manufacturing cleaning processes. We have wet cleaning and we have dry cleaning. Wet cleaning processes is using hot water that is sent through the piping as a way to clean and sanitize the piping. However, it is not used in manufacturing low moisture foods due to the risk of adding moisture to the equipment and potentially increasing contamination. Dry cleaning is another option. It is when you vacuum out the lines or push dry, dehydrated air through the lines. However, this is also not used in low moisture food processing, including peanut butter processing, because it does not remove the sticky residue off the equipment. So you still have to go in and somehow remove all the sticky residue that can occur when manufacturing peanut butter. So what's the best option for cleaning nut butter processing equipment? There is no GMP validated protocol for high-fat emulsified processes. The only option that we currently have is to use non-aqueous chemical solutions. However, more research is required to ensure that it works at the level as necessary to remove all potential contamination that can cause foodborne illnesses. So this research is out to determine if using a non-aqueous chemical cleaning and sanitization method involving 60% isopropanol with and without the addition of quaternary ammonia compounds on salmonella-contaminated peanut butter processing equipment is effective. So for this experiment, we set up bacteria that we had received frozen from the University of Georgia and the Minnesota Department of Agriculture. These salmonella strains that we received are the same salmonella strains that caused the 5 outbreaks of foodborne illness that we talked about earlier. We received them as frozen quanticolts, and after thawing, we added them to 10 mils of TSB broth. We then incubated that 10 mils for 24 hours at 32 degrees Celsius. We then took 0.1 mil out of the TSB broth and added it to a TSA-YE agar plate and let it incubate for 24 hours at 32 degrees Celsius. After the plate was incubated, it came out as a lawn culture, and we added 1 mil of TSB to the agar plate, and using a sterile L-shaped cell spreader, we dislodged the cell colonies from the surface of the agar. We then pipetted them up and got about 0.5 mils per plate. We harvested 40 plates of salmonella strains and combined 5 mils of each strain to make a salmonella cocktail, if you will. We then took the salmonella cocktail at a 1-to-1 ratio with peanut butter and added 1 mil of tween 80. That tween 80 is an emulsifier that allows the fat globules in the nut butter to mix with the liquid. Here's a diagram of the equipment that we used. You can see we have two steam-jacketed vessels. They are labeled oil tank and peanut butter tank, or T1 and T2. And we have stainless steel piping that connects through them all, going through a variety of simulated pumps that you would see when manufacturing peanut butter. We have a centrifuge pump, we have a positive displacement pump. All of the red circles that you see are environmental swab samples that we take. And as I go through the process, I'll explain to you when we take these environmental swabs. So the process will be conducted 3 times for each cleaning solution that we run. So it'll be 3 times for the 60% isopropanol and 3 times for the 60% isopropanol with quaternary ammonia compounds. So the first step in the process is to run hot soybean oil at 93 degrees Celsius into oil tank 1. We then run it through the system and discard until we see the oil is running clean. We do this to ensure there is no particles or contamination that is in the line that can disrupt our study. After it's running clean, we pump it back into oil tank, or T1, and then proceed to sample at all of the swab sample points that I showed you before, the 1 through 7, as well as take a microbial sample through a drain. We then autoclave creamy peanut butter until it reaches 55 to 60 degrees Celsius. This is done to ensure sterility as well as for ease of handling. We then add the peanut butter into tank 2, or the peanut butter tank, and allow it to circulate through the line. We discard the peanut butter until it comes out the consistency of peanut butter to ensure that the hot oil that was circulating before is out of the line. We then hook it up and circulate. Tank 2 continues to agitate as we add our contaminated peanut butter that we made earlier. We add the contaminated peanut butter and then allow the line to circulate for 15 minutes. During this 15 minutes, the line is kept at a temperature of 65 degrees Celsius using the steam vessel tanks. After 15 minutes, the peanut butter stops circulating and is sampled for microbial growth. We drain the peanut butter and then proceed to run room temperature soybean oil through the lines. This is to ensure that all clumps of peanut butter get emptied out before we start our sanitization and cleaning of this line. So the room temperature oil will circulate for 5 minutes, and then we will proceed to add 93 degrees Celsius soybean oil again for a hot clean process. Now this is the typical cleaning method currently used in peanut butter production. And for our experiment, we add the 93 degrees Celsius soybean oil, run it for 5 minutes, take a sample, and then continue to run it for 2 hours, sampling every 30 minutes for microbial growth. This is to determine if hot oil is successful in decreasing the log volume of salmonella. So after 2 hours, the hot oil stops circulating and is pumped back into the oil tank. The system is then left to sit for 24 hours. The point of this is that isopropanol's flash point is at 40 degrees Celsius, and for safety reasons, we wait 24 hours to ensure that all the equipment is cool enough to run isopropanol through. So at this point, we have cooled equipment, and we go ahead and run either isopropanol 60 or isopropanol with quaternary ammonia compounds throughout the system. We run the cleaning solution or the sanitizing solution through the system for 60 minutes, and then we go ahead and take swabs. For the results we got from this experiment, this table shows you the microbial survival in logs of CFU per gram of the salmonella in multiple different samples. We have it when we took samples when we first inoculated the peanut butter, and as you can see on our chart, that it was at 7.4 logs. And that is on TSA YE agar and XLD agar. XLD agar is a specific agar used for growth of salmonella, and TSA YE is a general agar. we took a sample after the initial oil flush. So we ran the peanut butter through for 15 minutes. Then we took the ambient temperature soybean oil, flushed it through for 5 minutes, and then we took another sample. And you can see there's a statistical difference in the microbial survival. It dropped about a log. We then took another sample after running the hot oil through for 5 minutes. We took a sample, and you can see once again there's a statistical difference between the initial oil flush and the hot oil after 5 minutes. So then the timer starts, and we take samples every 30 minutes for 2 hours. While the hot oil is running through, there is no statistical difference in logarithmic recovery of the salmonella. And that goes to show that the hot oil is not an effective measure against salmonella contamination. This slide shows the environmental swabs that we took. It shows all 7 sites that I showed you earlier. Site 7, is important to note, is a drain that does not come in contact with any of the product unless there's a pump failure. So you'll see Site 7 has no salmonella throughout the entire process. That is just going to show that the pump is functioning correctly and there is no contamination due to a malfunction of equipment. So before the experiment, we went ahead and took environmental swabs on all 7 sites, and as you can see, there's no salmonella detected. After the hot oil clean, so this is after we ran inoculated peanut butter through the system, we ran temperature oil through the system, we added the hot oil clean, and that ran for 2 hours, and then we took environmental swabs. You can see there's salmonella detected on 6 of the 7 sites, which means that we successfully inoculated the system, as well as proven that the hot oil clean is not effective in decreasing salmonella contamination to a safe level. The next sample we took was following the equipment cool down. So this was after the 24-hour period to ensure that the system was below the flash point of 40 degrees Celsius. Salmonella is detected in all 6 of the sites, again, showing that even after that hot oil clean, salmonella is able to thrive and proliferate throughout the system. The next sample we took was after the 60% isopropanol, and you can see that there is no salmonella detected on any of the 7 sites. The next sample taken was taken after the isopropanol with quaternary ammonia compounds. As you can tell and see, there is no salmonella that was detected on any of the 7 sites. The conclusions of this experiment are that the hot oil clean that is a current common practice in the manufacturing of nut butter is not effective in reducing salmonella to a safe point. Our detection limit was 0.16 log per CFU per gram, and the hot oil cleaning and the 24-hour cool down period were not enough to eliminate the salmonella. Our experiment does show that isopropanol 60% and isopropanol with quaternary ammonia compounds were both effective in reducing salmonella beneath our detection point. This is an important study because it shows that the non-aqueous chemical solutions, especially isopropanol 60 and isopropanol with quaternary ammonia compounds, are effective option that we could study further and potentially make into a regulated cleaning and sanitization process for low moisture foods. I would like to acknowledge and express my sincere gratitude to the University of Georgia and the Minnesota Department of Agriculture for the salmonella isolates, as well as to my co-writers. Are there any questions? Thank you.